Gametogenesis. Gametogenesis is the process by which gametes are formed in organisms. It ensures that offspring inherits genetic material from both parents, contributing to genetic diversity. It includes spermatogenesis and oogenesis, both of which are crucial for sexual reproduction, ensuring the correct number of chromosomes in offsprings.
Spermatogenesis, the process by which primitive germ cells in the seminiferous tubules of the testis are converted into mature haploid spermatozoa. This process begins during adolescence and continues throughout life. It is divided into 3 stages. Spermatocytogenesis. Male germ cells undergo mitotic division and differentiate to form type A, dark and pale, spermatogonia. The pale spermatogonia undergo mitosis to form type B spermatogonia, while the type A do not undergo further division. The type B spermatogonia undergo differentiation to form primary spermatocytes. Meiosis. The primary spermatocytes undergo the first meiotic division to form 2 secondary haploid spermatocytes. These secondary spermatocytes undergo the second meiotic division, forming spermatids. These spermatids are further converted into spermatozoa by the process called spermiogenesis. Oogenesis. Oogenesis is the process of ova formation from primitive germ cells in the ovary beginning during fetal life. Primitive germ cells multiply mitotically to form oogonia. The oogonia further differentiate to form primary oocytes. Each primary oocyte is enveloped by a single layer of granulosa cells forming a primary follicle. A homogeneous membrane, the zona pellucida, appears between the follicular cells and the oocyte. The primary oocyte is arrested in prophase 1 of meiosis during intrauterine life, persisting until adulthood. This arrest occurs due to the secretion of oocyte maturation inhibitor. Further development occurs with the onset of puberty. The primary follicle is further surrounded by another layer of granulosa cells, forming a secondary follicle. An antral cavity now appears between the follicular layers. The antral cavity enlarges, filling with antral fluid, and the granulosa cells differentiate to form the theca interna, which secretes estrogen, and the theca externa. This secondary follicle matures into a Graafian follicle. Within the Graafian follicle, the primary oocyte completes the first unequal meiotic division, forming a secondary oocyte, which is arrested at metaphase by the protein pp39 M.O.S. The secondary oocyte receives most of the cytoplasm, and a small polar body, which typically degenerates, is also formed. With the formation of the secondary oocyte, ovulation takes place. If the sperm and the secondary oocyte reach the ampulla, fertilization and the second meiotic division complete simultaneously through the destruction of pp39 M.O.S. by calpain, forming a mature ovum. In the absence of fertilization, the ovum degenerates. Thus, from one primary oocyte, one haploid ovum is formed.